Today I will talk about horizontal gene transfer events in pox viruses. And pox viruses are large double-stranded DNA virus. Oh, sorry, So pox viruses are large double-stranded DNA virus which belongs to the family Poxviridae. And the genome size of pox viruses varies between 136 to 360 kb for different pox viruses. And the pox virus as a family has very variable host range. Some members of the pox virus can infect various host species, other can infect very limited number of hosts. And pox viruses like other DNA viruses acquired large number of genes that are homolog of cellular genes, which pox viruses might have acquired through horizontal gene transfer. And horizontal gene transfer is the transfer of genetic material between different organisms. An example can be gene transfer between viruses and eukaryotes. In nature it is very widespread, and the horizontal gene transfer can play a role in shaping functional diversity and evolution of viruses or organisms. And the predicted acquired genes of pox viruses include interleukin 10, TNF alpha receptor and apoptosis inhibitor, and some other genes also. And the function of those genes ranging from cell cycle control to facilitating the host immune evasions. But it is not known how the viruses acquired those host genes, since pox viruses exclusively replicate in the host cytoplasm in the viral factory, and there are no experimental evidence of horizontal gene transfer in pox viruses. There are two possible routes for gene transfer. It can be either a direct DNA integration, or it can be an RNA dependent mechanism by reverse transcription and integration of the host genes into pox viruses. The objectives of this study includes detection of horizontal gene transfer in an experimental system and to characterize the molecular mechanism of horizontal gene transfer. To efficiently detect the horizontal gene transfer event, we have designed an experimental system by harnessing the host antiviral protein pKR pathway, where pKR can sense the double-stranded RNA produced during virus infection. pKR binds to double-stranded RNA and homodimerases, and which leads to its activation. These activated pKR then phosphorylate the translation initiation factor EF to alpha, which leads to the suppression of general mRNA translation, and thus pKR activation inhibits the virus replication. However, viruses have immunomodulatory genes that can inhibit this pKR activation. E3L and K3L are pKR inhibitors from pox viruses. So E3L can inhibit this pKR activation by preventing pKR homodimerization, and K3L can inhibit by preventing pKR and EF to alpha interaction. Thus E3L and K3L inhibit pKR and prevent the suppression of general mRNA translation and allow virus replication. So for my experimental setup, I used this vaccinia virus E3L as a selective marker, and EGFP expressing vaccinia virus that lacks both this E3L and K3L pKR inhibitors. So this parental virus cannot replicate in the wild type cells, as they cannot prevent or inhibit the pKR, but they can replicate in the permissive cell lines, the cell lines that has the pKR knocked out or stably express the E3L or K3L. And for our experimental design, we want to detect if this parental virus can acquire E3L to replicate in the wild type cells. So for my experimental setup, I used a construct which contains SB40 promoter and intron to detect if the gene transfer is DNA or RNA mediated, and vaccinia virus synthetic early late promoter followed by red fluorescent protein mCherry fused with the pox virus E3L. So I stably transfected rabbit kidney cell line RK13 wild type. Now the RK13 cell line are expressing this mCherry E3L. These cells are then infected with the parental vaccinia virus that lacks E3L and K3L. So this virus cannot replicate in the wild type cell lines, but they can replicate in this E3L expressing cell line. After the initial infection, we collected the viruses and infected the wild type cell lines. these viruses will only replicate in this wild type cell lines if they acquired the M-cherry ETH from this host. And those viruses genome and the integration site of the ETH will be analyzed to characterize the molecular mechanism of horizontal gene transfer. And those viruses will form plaques that will express both GFP and M-cherry. And for the first time, we detected the horizontal gene transfer experimentally in the pox viruses. And so far, I have identified 11 vaccinovirus isolates that took up the M-cherry ETH from their host. And you can see the integration sites in the pox virus genome throughout the genome here. And we have also calculated the frequency of horizontal gene transfer for the M-cherry ETH in RK13 cells, which is 1 in 35 million infectious virions. And this is the clone 1, my very first clone. And here you can see the construct I used to make the stable cell line to compare it with the integration site. So in my clone 1, M-cherry ETH was integrated into the intergenic space between H4L and H5R of pox virus. And integration site revealed that this intron was spliced out and there is a poly-A region after the ETH shell, which indicate that this gene transfer is an RNA-mediated gene transfer. And the target site at both ends were duplicated, which is the signature of retrotransposon activity. And in my clone 3 here, however, the M-cherry ETH was integrated into the H4L, which is a RNA polymerase subunit and an essential gene. And this also consistent with the RNA-mediated gene transfer and retrotransposon activity. However, we are so surprised that how this clone 3 can even replicate if the M-cherry ETH is integrated into an essential gene. So to address this question, I compared my clone 3 and clone 1, and you already know that in clone 1 it is in the intergenic space. It didn't integrate in any gene. So I PCR amplified this integration site, and as is expected, you can see the larger band is the indicating that M-cherry ETH integration, and the smaller band in the control is the intact site here. In comparison, in the clone 3, where it is integrated into an essential gene, you see the PCR amplification yielded 2 distinct bands in clone 3, where this larger bands indicating the M-cherry ETH integrated H4L, but this smaller band is indicating that there is a intact copy of H4L, which might explain that how this clone 3 can replicate in the if it is integrated into an essential gene. So there are 2 possible scenarios that can explain this intact H4L. It can be that the viruses has the duplicated H4L, or the coexistence of the parental virus and my clone, and they can complement each other in the cells, where the clone 3 supplies the M-cherry for the parental virus, and parental virus can supply the intact copy of H4L. So to address the question if the complementation is a possibility, I infected this clone 3 in a permissive cell line, where which is the RK13 cell that stably express ETH and K3L. And this is permissive for the parental virus. So here you can see that most of the clones are green, have green plaques, and some of them are M-cherry ETH expressing plaques. So this might indicate that this clone 3 has parental virus in it, which might indicate that there might be complementation going on. So after that, we also confirmed the parental virus by long-range sequencing, and after that I serially passed this clone 3 to analyze the fate of the population in clone 3. And I have serially passed this clone 3 at low MY in RK13 wild type cells, and after 17 passages I did the PCR amplification of this H4L. So as I mentioned earlier, that there are 2 possibilities which might explain the intact copy of H4L. The other possibility was the duplication of H4L. So I PCR amplified the H4L locus with outward primers. So if there is a duplication of the H4L, we would see like PCR amplify these breakpoints of this H4L. And here you can see that we found the fragments in the PCR amplification after passages, but not in the initial clone 3, which might indicate that the duplication happened after the complementation. And we also confirmed by sequencing, and we saw that this H4L was duplicated in the neighborhood of this intact H4L. So to summarize, we successfully detected the virus took up the E3L from the host cell, and so far I have isolated 11 clones that took up the E3L, and all of them indicated that RNA-mediated gene transfer and retrotransposon activity. And the gene can have two fates. If the integration happened in a non-essential locus, it can replicate by itself, or if the integration happened in an essential gene, the parental virus can complement each other with the clones, and after passaging, these viruses can recombine and it can duplicate the essential gene. And in this slide, you can see the three important ways pox viruses can do the evolution. So with that, I would like to thank Dr. Stefan Rothenberg, Dr. Greg Brannan, our previous lab member Sherry Holler, and Rothenberg lab members. Thank you.
Okay, we have a question in the back there. We have a
Hey, so your hypothesis is that it's a retrotransposon that is like helping to integrate. So did you try to do the same experiment but with a reverse transcription inhibitors to see if you don't see the same type of integration?
Can you please repeat the
Did
question?
you do the same experiment but with a reverse transcription inhibitor?
Reverse so yeah. So you mean the inhibitor of reverse transcription?
Yes, because if you say that it's a transposon that is helping to integrate the RNA.
Actually what we did, we transfected with the line 1 element, which is a retrotransposon. So it actually works like a target prime reverse transcription. So retrotransposon bind with the mRNA and then make the cDNA and integrate at the same time. So with the line 1 transfection, we actually found clones, but we are not sure if the reverse transcription, the retrotransposon did that. But we saw the signatures of retrotransposon activity here.
Okay, we have one more question. Okay.
Is there any evidence that poxvirus infection enhances the expression or activity of the line elements?
Sorry, I didn't understand.
Is there any evidence that poxvirus infection stimulates lines, their expression or activity?
Actually, line 1, the stimulation of line 1 can happen during the stress also. So, I mean, I'm not sure, because poxvirus infection can also like stimulate, maybe stimulate, but because it can produce the stress in the cells. So that is a possibility also.